Episode 2, Ribavirin and RNA viruses, Molecular Warfare. Welcome back to Ribavirin Reminiscence and Lingering Load in Long COVID-19, where we splice through the molecular saga of an antiviral veteran. I'm your host Anushka with a master's in genomics and a knack for unraveling the genetic threads of life. After tracing ribavirin's journey from the 1970s to hepatitis C in episode 2, today we are zooming into its molecular battlefield. How does this drug outwit RNA viruses and why is it dubbed a mutagenic wrecking ball? Joining me is Saakshi, a master's student in microbiology whose insights into microbial warfare glow like a bioluminescent colony in a dark lab. Welcome, Saakshi.
Thank you, Anushka. I am thrilled to dive into the molecular mechanics of ribavirin and its war on RNA viruses.
Let's sequence the basics like aligning a critical genome. Ribavirin is hailed as a nucleoside analog. What does that mean and how does it set the stage for its antiviral action?
A nucleoside analog is a synthetic molecule engineered to resemble the natural nucleosides, which are adenosine, guanosine, cytidine, or uridine, that forms RNA or DNA. Ribavirin mimics guanosine with a triazole ring tweaking its structure to deceive viral enzymes. Once inside cells, host kinases phosphorylate ribavirin into its active triphosphate form, or what is known as RTP, which has a half-life of nearly 24 hours. This RTP is the warhead. Viral RNA-dependent RNA polymerases, or what we call as RDRPs, mistakenly incorporate it into nascent viral RNA strands, disrupting the process of replication. Unlike host polymerases, which have proofreading, viral RDRPs are error-prone, hence making them vulnerable to ribavirin's sabotage. This mimicry is the cornerstone of its ability to infiltrate and destabilize viral genomes.
A cunning imposter in the viral assembly line. Summarizing, ribavirin's guanosine mimicry tricks viruses into faulty replication. How does this lead to the chaos that cripples RNA viruses?
Ribavirin's chaos unfolds through a multi-pronged assault tailored to the virus. Its primary weapon is the lethal mutagenesis. RTP incorporation into viral RNA induces G to A or C to U transitions, spiking mutation rates. Example, 10 raised to the power minus 3 errors per nucleotide in HCV. This pushes viruses beyond their error threshold, hence producing defective, non-infectious progeny, where studies show a 90% viability drop in RSV. Secondly, ribavirin inhibits inosine monophosphate dehydrogenase, or what is known as IMPDH, slashing GTP pools by 30 to 50%, starving RDRPs of raw materials, critical for viruses like Lassa fever. Thirdly, in some cases like influenza, RTP directly binds RDRP, hence blocking elongation at an IC50 of approximately 10 micromolar. This versatility of mutagenesis, resource depletion, and polymerase jamming makes ribavirin a molecular Swiss Army knife, disrupting replication across diverse RNA viruses.
A triple threat saboteur dismantling viral machinery. Summarizing, ribavirin's mutagenesis, GTP starvation, and polymerase inhibition wreaked havoc. Why does its broad from activity captivate researchers?
Ribavirin's broad-spectrum prowess stem from targeting conserved vulnerabilities in RNA virus biology. Unlike antivirals like oseltamivir, which homes in on influenza's neuraminidase, ribavirin exploits universal processes, RNA synthesis and nucleotide metabolism as well. Most RNA viruses, such as HCV, RSV, and Lassa, rely on error-prone RDRPs, where the error rates merely are from 10 raised to the power minus 4 to 10 raised to the power minus 5, and lack proofreading, make them susceptible to ribavirin mutagenesis. IMPDH inhibition affects all GTP-dependent viruses from flaviviruses to arenaviruses. In vitro screens like those in 1972 ICN studies showed ribavirin active against 80 plus RNA viruses with the EC50 values ranging from 1 to 100 micromolar. This universality, rare in antivirals, allows ribavirin to tackle diverse pathogens from hemorrhagic fevers to respiratory infections, making it a go-to in emergencies like Crimean Congo outbreaks.
A universal key unlocking viral weaknesses. Summarizing, ribavirin's focus on shared RNA processes fuels its wide reach. How does it compare to newer nucleoside analogues like remdesivir or sofosbuvir?
Ribavirin, remdesivir, and sofosbuvir are nucleoside analogues, but their strategies are diverge. Specificity, let's talk about that first. Sofosbuvir target HCV's NS5B polymerase, where the IC50 is nearly 0.1 micromolar, and remdesivir hits SARS-CoV-2's RDRP, achieving 99% inhibition at 1 micromolar. Ribavirin's broader action, where the EC50 is nearly 10 to 100 micromolar, is less potent, requiring higher doses. Let's talk about mechanism. Ribavirin drives mutagenesis, increasing HCV mutation rates nearly fivefold, while remdesivir and sofosbuvir cause chain termination, hence halting RNA synthesis mid-strand, as seen in 95% replication block as in SARS-CoV-2. Let's talk about toxicity. Ribavirin's IMPDH inhibition disrupts host GTP pools, causing hemolytic anemia, which is seen in nearly 15 to 20% incidence. Newer drugs with tighter viral specificity reduce host effects. Talking about remdesivir's hepatotoxicity, it is nearly 5%, and ribavirin's generalist approach suits polyviral threats, but targeted snipers like sofosbuvir dominate single virus diseases.
A veteran warrior versus precision sharpshooters. Summarizing, ribavirin's broad mutagenic approach contrasts with other drugs' specificity and lower toxicity. Can viruses outsmart ribavirin through resistance?
Resistance is possible but complex due to ribavirin's multi-modal attack. Single-point mutations in RDRP like HTV's Y415F reduce ribavirin's incorporation, lowering mutagenesis by 30%. But these are rare, as multi-target hits, RDRP, and IMPDH demand multiple mutations. Intriguingly, some viruses like poliovirus evolve high fidelity RDRPs, hence reducing error rates by, let's say, 2 to 3-fold to dodge lethal mutagenesis, essentially. They proofread better, as seen in 2010 Nature studies. This fidelity shift, however, shows replication by 20% hampering fitness. Resistance is thus less common than single-target drugs like sofosbuvir, where NS5B mutations emerge in 5 to 10% of HCV patients, hence making ribavirin's broad action a tougher barrier for viral evolution.
A viral chess game where ribavirin keeps viruses in check. Summarizing, resistance via polymerase tweaks or fidelity is rare but possible. Saakshi, what's the key takeaway about ribavirin's molecular warfare?
Ribavirin is a molecular mastermind, wielding lethal mutagenesis, GTP depletion, and also polymerase inhibition to cripple RNA viruses from RSV to HCV. Its broad spectrum strength is it's active against 80% viruses, stems from targeting universal RNA processes, unlike the sniper-like precision of remdesivir or sofosbuvir. Though less potent and more toxic, nearly contributing to 15% anemia risk, its multi-pronged attack limits resistance, offering lessons for antiviral design. As we explore its role in long COVID, where viral persistence may linger, ribavirin's chaotic genius remains a vital tool, reminding us that even old warriors can fight new battles.
Sakshi, you have mapped ribovirin's molecular strategy like a genomicist charting a viral variant. Thank you for this electrifying dive.
My pleasure, Anushka. It's been a blast dissecting ribavirin's tactics with you.
Next week on ribovirin reminiscence and lingering load in long COVID 19, we'll probe viral persistence in long COVID and whether ribovirin mutagenic might shift the tide. I'm Anushka, and this is your molecular deep dive. Stay safe, stay curious.